Everyone. How are you all today? Good. Anybody do anything fun? Hockey game? Anyone go to the hockey game? No. This is why that stadium is, or the new arena is half empty for the games. You guys have to, yeah, think about going to the hockey game. Okay. I didn't go either, so I guess I'm part of the problem. But if you ever go to a hockey game, you can have a drink at the Duluth Curling Club beforehand and tell them that you're a personal guest of Dr. Skildum and they'll let you in. Okay, what else? Heck of the North, that was in town this weekend? Anybody do that? No? Okay. What else? Fall Color? Apples in Bayfield? Is that this weekend? Next weekend? Okay. All right. A lot of distractions, huh? Okay, well, topic for today is amino acid metabolism. So amino acids, we've touched on them, towards the first weeks in class. We're going to hit them a little bit more today. And we'll also consider them, they're integral to the urea cycle that we'll talk about on Wednesday. And they're also integral to the vitamin B12 metabolism and tetrahydrofolate metabolism that we'll talk about next week, I think. Okay, so we'll kind of continue to fill out the amino acid metabolism story as the course unfolds. Because of the chemical diversity of the side chains of amino acids and their multiple uses in the cell, they don't fit neatly into a single lecture. That said, what we're going to talk about today, we'll talk about basically how we can synthesize the amino acids that we can synthesize and how we can degrade those amino acids and also to degrade amino acids that we can't synthesize. Okay, and once we have the normal biochemical pathways introduced, then we can talk about some cases where those pathways are disrupted and hopefully link some of the symptoms of the pathologies to the biochemical defects. Sound good? Okay. As I'm looking at these objectives, there's one, the homocystinemia, or this is sometimes called hyperhomocystinemia. We might touch on this one, but this is really kind of belongs with the tetrahydrofolate B12 lecture. So yeah, put a pin in this one if it's not clear at the end of this lecture, and it will be in the future. Okay, let's see. Should we start with a little clicker quiz? Oops. This is a nice short stem, but it's a good two-step question, right? Gotta recognize the cofactor as pyridoxal phosphates, vitamin B6, and remember that lean meats, I'm not certain that it's absent from those other food sources, but lean meats would be the best source of it among those choices. Okay. think we introduced the thymidine kinase and the thymidine synthetase enzyme and the thymidine nucleotide cycle a long time ago. Okay, best answer is serine. So serine is the most important donor of carbon to the one carbon pool. Okay, so we'll talk about serine and how that's synthesized and degraded today, and we'll talk about its role in one carbon metabolism later on in the course. Okay, so remember the side chain of serine is the alcohol, single carbon alcohol. And if you take away that carbon, then you're left with nothing on the alpha carbon. And that nothing on the alpha carbon, that means that you've got the amino acid glycine, right? Okay, very good. Okay, so just to warm us up there, a little brain teasers. Okay, to orient you where we are on the metabolic map, we're roughly in the blue section for today. And we're dipping into the TCA cycle and into and out of glycolysis. Okay, so hopefully we'll be treading some somewhat familiar ground. In terms of the higher order purpose of amino acid metabolism, amino acid metabolism allows us to store excess amino acids as protein, and that's through translation. That's more of the bailiwick of Dr. Cormier. We can also use amino acid catabolism to get energy from amino acids. Okay, so that's kind of big overview. Okay, in terms of amino acid metabolism, the synthesis, the transport, and the catabolism of amino acids are all enzyme-mediated reactions. And those enzymes use non-protein cofactors, as we discussed way back in like week 1 or week 2. So amino acid metabolism, we'll see some examples, at least one example of thymine pyrophosphate participating in a decarboxylation reaction. But really, like the most important amino acid cofactor is the vitamin B6 derived pyridoxal phosphate. That reactive site here, that is just the perfect fit to bind into a peptide backbone, and it allows enzymes to grab onto an amino acid, and then other parts of the amino acid of the enzyme can do some chemical reaction on it. Okay, so pyridoxal phosphate, this is, I think, the most important amino acid cofactor, not exclusive, certainly. Let's see, some new ones, I guess tetrahydrofolate, this is important for that methionine cycle, and the thymidine synthetase cycle, and one carbon metabolism. So I'm going to touch on this one, and we'll elaborate on it more next week. One that we haven't seen at all, I don't think, is tetrahydrobiopterin, and this is a redox cofactor that participates generally in life in ring hydroxylation reactions. In the context of human biology, its importance is as a cofactor for the synthesis of tyrosine from phenylalanine. So phenylalanine hydroxylase uses tetrahydrobiopterin as a redox cofactor. Okay, and tetrahydrobiopterin is not a vitamin-derived redox cofactor, whereas the pyridoxal phosphate, this is from vitamin B6, and the tetrahydrofolate is from folic acid. Okay. Okay, so just some general introduction. When we talk about the enzymes that um, we are able to synthesize ourselves, or the non-essential amino acids, um, when we synthesize them, we use certain enzymes, and when we catabolize them, we use a different set of enzymes. So, like with glycolysis and gluconeogenesis, for example, those two pathways that are opposites of one another, they're using a lot of the same enzymes for many of the steps. Right? Not the key steps, but many of them are the same. And that's not the case with amino acid synthesis and amino acid catabolism pathways. Generally, those are using totally different enzymes. Okay? With the non-essential amino acids, or the amino acids that we can make ourselves in the absence of any protein in our diet, those can be synthesized from either intermediates of the TCA cycle or intermediates of glycolysis. Likewise, when those amino acids or our essential amino acids are degraded or used as fuel sources, they can either be degraded into intermediates of glycolysis or intermediates of the TCA cycle. Okay, and when we consider the degradation, amino acids can be either gluconeogenic or ketogenic, and I'll elaborate on that concept shortly. Okay, so in terms of the essential amino acids, or the amino acids that we don't have the capacity to synthesize, they are shown here. So we know we can characterize amino acids based on their chemical structure of the side chain or their hydropathy index, or we could alphabetize them if we want. So whether they're essential or non-essential in our diet is another way that we can categorize them. So the essential ones, the MV Pithal, I don't know if you remember that from undergraduate days, but that's acronym for the, or a acronym, a mnemonic. Yeah, mnemonic for the essential amino acids. Some amino acids, now this list is generally accepted to be the essential amino acids, but you have to consider the context, I guess. Some amino acids are conditionally essential. For example, arginine, if you are not gaining mass, generally we have the capacity to synthesize enough arginine for an adult who's not, trying to gain mass through bodybuilding or something like that. So arginine is a essential amino acid for children, but it's not considered an essential amino acid for adults. Likewise, some amino acids, the ones that we're able to synthesize, for example, tyrosine being synthesized from phenylalanine, if we don't have phenylalanine in our diet, then tyrosine is an essential amino acid, right? Or if we have phenylketonuria deficiency in the phenylalanine hydroxylase enzyme, where we don't have the capacity to do this reaction, then tyrosine would become an essential amino acid, even though it's not on the MV Pithal list. Okay, does that concept make sense? Then, yeah, just another example that we'll see later in the lecture, cysteine biosynthesis, that we're able to synthesize cysteine, but it's dependent on a sulfur from methionine. So if we didn't have methionine, or if we didn't have enough methionine, then cysteine would become an essential amino acid. Okay, so just some examples there. Okay, so amino acids can be derived from, or they can degrade into either glycolysis intermediates, TCA cycle intermediates, or the ketone bodies, acetoacetate or acetyl-CoA. So we kind of lump together acetyl-CoA and acetoacetate. Acetoacetate can be split into two acetyl-CoAs. So we'll consider them in these orders, or in this order, and talk about the glycolytic intermediates first. So the most simple is the transamination of pyruvate to alanine. And this one we'll discuss this, I think, at the last one. The first one that we'll discuss is the derivation of serine from the triose phosphate or the phosphoglycerate step, or excuse me, phosphopyruvate step. Let's see. So just to round this out, so we can get alanine directly through a transamination reaction of pyruvate. The serine, this can be derived from glycolytic intermediate, and then serine can be used to synthesize glycine through that loss of a carbon to the tetrahydrofolate reaction. Serine can gain a sulfur from methionine to become cysteine, and then serine can degrade back into the glycolytic intermediates. Okay, so that's where we're going, and we'll talk about that generation of serine first. So we have the glycolytic pathway up here. So glycolysis generates the glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Those are going to be converted ultimately into the 3-phosphoglycerates. This then is mutase to the 2-phosphoglycerate, converted into the phospholynyl pyruvate through the hydration, and then the pyruvate kinase can ultimately generate pyruvate. So normal pathway of glycolysis would be here. Serine biosynthesis can dip off of the 3-phosphoglycerate, and serine degradation can generate the 2-phosphoglycerate. So let's look at the condition of serine degradation first. This would be the case of, say, if you were catabolizing protein, either because you had a very high protein diet or a high protein meal in the fed states, or if you were doing muscle protein breakdown and catabolism in the fasted state, you might have serine around that you wanted to convert into a carbon dioxide and gain energy from it. Okay, so the first step in serine catabolism is a transamination or a removal of the amine group to make the keto acid hydroxypyruvate. The ketone is then reduced to an alcohol to make glycerate. The glycerate then is phosphorylated by the phosphoglycerate kinase using ATP as a phosphate donor to make the 2-phosphoglycerate. So these basic steps are going to be recapitulated in the biosynthesis of serine, but in a reverse direction, I guess, if you will, and by different enzymes. Okay, if we take the counter situation where we don't have enough serine and we wanted to synthesize it from a glycolytic intermediate with the 3-phosphoglycerate as the substrate. So here first the alcohol is converted into a ketone, then we get a transamination reaction to make the first amino acid, and then the phosphate is removed by a phosphatase as inorganic phosphate to make serine. So this reaction, because we're taking one of the phosphates of our glycolytic intermediate and we're making inorganic phosphate, but we're not making an ATP, this is, I guess, less energy generating potential than if we just went on the north side of the map, just down glycolysis. Okay. Nevertheless, if we make serine from that glycolytic intermediate, we can then use it for diverse purposes. We could use it for protein synthesis just through the process of translation. If it was a cell that needed a lot of nucleotides or other biosynthetic reactions that required a single carbon transfer, that serine might be converted into glycine through and to support tetrahydrofolate metabolism. So yeah, this serine, once it's being synthesized here, it could support diverse purposes. One of the diverse purposes that it could serve is shown here, the donation of carbon to tetrahydrofolate, and that is, we'll talk about that tetrahydrofolate business and elaborate on that at length in the B12 tetrahydrofolate lecture. So that's one side of it, but for the purposes of the amino acid metabolism lecture, this reaction is how we could synthesize glycine. Okay, so glycine is not essential in our diet. If we need it, we can synthesize it from serine by removing that alcohol carbon side chain and giving it to tetrahydrofolate. So glycine then, this has different metabolic fates. We could use it as a substrate for protein translation if we were making protein. We can also catabolize it down to TCA cycle intermediates. We can generate that glycine in addition to serine catabolism. The amino acid threonine can also be substrate for glycine biosynthesis. This is a through the elimination again of the 2-carbon alcohol in this case. So it's kind of a similar reaction to the serine breakdown, but in this case we're doing a 2-carbon alcohol, and that's being relieved as acetaldehyde. So this acetaldehyde, this could be then converted into acetate by acetaldehyde dehydrogenase and make acetyl-CoA essentially. So this is like a easy to use common intermediate in a lot of different pathways. Let's see, so glycine, this could have multiple metabolic fates. The glycine cleavage enzyme, this is kind of breaking it down into its, most simple bit. So this is making carbon dioxide and ammonia, and then taking the last carbon and adding it to tetrahydrofolate, again through a single carbon reaction, to make the N5-N10 methylene tetrahydrofolate. So this would be a pathway if we wanted to get the maximum energy and recover the carbon from glycine catabolism. Okay, so that's one metabolic fate of glycine. The glyoxylate pathway is another metabolic fate of glycine, and this is either a transamination where the amine group is donated to a keto acid pyruvate to make alanine, or a oxidative deamination to make free ammonia. Glyoxylate, this then can be converted into a TCA cycle intermediate by reacting with alpha ketoglutarate. This makes the alpha hydroxy ketoadipate, and structurally this is a 6-carbon. It's not a tricarboxylate, it's a dicarboxylate, but it's similar enough to citrate or isocitrate that it can be catabolized in the TCA cycle down to carbon dioxide. So glyoxylate can be eliminated completely down to carbon dioxide through becoming a TCA cycle intermediate. It has this other pathway of becoming this very simple dicarboxylic acid oxalate. This can be excreted in the urine, so it's got the two carboxylic acid ends, so it's quite soluble. However, if the synthesis of oxalate is very rapid and is exceeding the capacity to pee it out, this can condense through like making ionic bridges with other salts and form kidney stones. So this is kind of a long pathway to get to the pathological significance. So kidney stones just generally are one of your objectives, and you'll get a ton of nephrology in year 2, but we'll just so don't go too deep into it. But the cystinuria is a specific kind of kidney stones that we'll talk about later in the lecture. And then oxalate, this could be kidney stones based more on your diet and hydration status rather than inborn error of metabolism like cystinuria would be. So anyway, just don't go too deep into kidney physiology or pathology, but just kind of know a little bit about it. Okay, let's see, where were we? Okay, so we were talking about amino acids that could come from glycolytic intermediates, and serine is kind of the most proximal to glycolysis, right? So we can get glycine from serine. We can't get threonine from serine, but we can get threonine into glycine and then serine from glycine. Okay, so we can also get synthesis of one of the two sulfur-containing amino acids, cysteine, from serine. This is a reaction where the serine first makes a bond through the sulfur of homocysteine, then the carbon portion of the homocysteine is cleaved off and the sulfur portion remains. So homocysteine, this is generated from methionine, and this is part of the lecture where the tetrahydrofolate cycle, this is kind of up here somewhere. But methionine is like homocysteine if it had a methyl group on top of the sulfur here. Okay, so when methionine gives off that single carbon group, then homocysteine is left, and homocysteine can, be methylated again to make methionine. But another metabolic fate of the homocysteine is to bond to the serine through that sulfur group, and then the carbon part of it is cut off, and then what's left is the free sulfur, except now it's on the cysteine amino acid. So this carbon portion that's remaining, and this enzyme is kind of an unusual enzyme in that it's doing that cleavage of the sulfur bond, and then it's also deaminating the large amalgamation of the homocysteine and the serine, and releasing the releasing free ammonium. So the carbon portion that's cut off here, this can be converted into the TCA cycle succinyl-CoA through that same pathway that is used to generate TCA cycle intermediates from a branched chain fatty acid or from an odd chain length fatty acid. Do you remember that? So there's propionyl-CoA carboxylase, makes methylmalonyl-CoA, and then methylmalonyl-CoA mutase makes succinyl-CoA. The first one needs biotin, the second one needs vitamin B12. Okay, just biochemical trivia there. Okay, so this is a pathway of serine being converted into the amino acid cysteine. Generally, we wouldn't be making cysteine from serine just to burn it or use it in a catabolic reaction, but just to complete the picture, we can convert we can convert cysteine into pyruvate if we were catabolizing cysteine. So that's first in the catabolism of cysteine. It is first the sulfur is first. The free sulfhydryl is converted into a sulfite. Then the transamination reaction removes the amine group, and then pyruvate plus the sulfite is left. The sulfite goes away as inorganic sulfur or sulfite. The sulfite can either be urinated out, or there's this molecule called activated sulfur or PAPS, and the sulfate can be used for the synthesis of this PAPS molecule. The PAPS, or the 3' 5' phosphoadenosyl pyrophosphate, this something like that. It's written out here. Anyway, it's a nucleotide that contains phosphates in a high energy bond and also sulfates in a high energy bond. So similar to like ATP being a phosphate donor, this is a sulfate donor, and it's important for the synthesis of like chondroitin sulfates in proteoglycans. And so anyway, it's a biosynthetic sulfate donor. Okay, so I think that is then rounding out the amino acids that can be derived from serine through from glycolytic intermediates. Then the final one is the simplest one, the transamination of pyruvate to alanine. So alanine aminotransferase, this is using alpha-ketoglutarate and a glutamate as a keto acid acceptor and an amino acid donor for the aminotransferase reaction. So this then could make a nice segue into amino acids that are derived from TCA cycle intermediates, because we can get the amino acid glutamate from the TCA cycle intermediate alpha-ketoglutarate through the same reaction. Does that make sense? Okay. All right. So are there any questions about those alanine or serine-derived pathways? Okay, let's carry on then. So the amino acids that we can get from the TCA cycle intermediates are either going to be derived from oxaloacetate, and those are going to go through aspartate, and then asparagine can be synthesized from aspartate. Glutamate, then the transamination of or oxidative deamination of glutamate can make alpha-ketoglutarate, and we can also generate glutamine through addition of another amine group, or arginine or proline from glutamate. Okay, so basic biosynthesis of that is shown here. So let's consider the oxaloacetate-derived amino acids first. So aspartate aminotransferase, this is very similar to alanine aminotransferase in that we're getting a amine transfer from the keto acid oxaloacetate to make the amino acid aspartate. So not shown in this diagram is the transamination partners. In this case, the aspartate aminotransferase, like alanine aminotransferase, is using the alpha-ketoglutarate as an acceptor and glutamate as an amine donor. So kind of this part of the TCA cycle intermediates are present in multiple different reactions. Okay, so once we have oxaloacetate from or excuse me, once the transamination of oxaloacetate generates aspartate, then this can be used for protein synthesis if it's needed. We can also get the amino acid asparagine from aspartate, and this is a kind of multi-step reaction that's using ATP hydrolysis to provide energy and the amino acid glutamine to provide nitrogen to generate this new nitrogen carbon bond to make asparagine. Then in the catabolic sense, we can go back from asparagine into a TCA cycle intermediate through the asparaginase reaction. Again, just illustrating this idea of the catabolic pathways and the synthesis pathways using totally different enzymes. The asparaginase, this is cleaving off that same nitrogen as free ammonium, so we're not gaining anything. We're not generating glutamine, for example, from glutamate by going back through this pathway. Okay, so the oxaloacetate-derived amino acids, getting aspartate and asparagine is pretty straightforward. Let's consider the ones that go through or that are derived from alpha-ketoglutarate. So alpha-ketoglutarate, this can be converted into glutamate through two different pathways. One of them we've talked about, that transamination pathway, and this could use either the alanine aminotransferase reaction or the aspartate aminotransferase reaction, or there are diverse other aminotransferase reactions that are using another keto acid to make an amino acid, and they all use alpha-ketoglutarate and glutamate as partners. So we can get the glutamate here through diverse reactions, just the AST is shown as an example. There's another different way that we can get glutamate from alpha-ketoglutarate, and this is a somewhat rare reaction in the body, the formation of a new nitrogen carbon bond using ammonia or ammonium, as a substrate. So there are a lot of reactions in our body, enzymes that can take nitrogen from an amino acid or some other substrate and shuffle around a pre-existing carbon-nitrogen bond, but there are very few enzymes that can make a new carbon-nitrogen bond using inorganic nitrogen as a substrate. So glutamate dehydrogenase is one of them. And glutamate dehydrogenase, this is catalyzing the oxidative deamination, or it's also a reversible reaction, so we can get the amination of alpha-ketoglutarate to make glutarate. So the fact that glutamate dehydrogenase is a nitrogen-fixing enzyme in our body, that's kind of one unusual feature of it. Another unusual feature of the glutamate dehydrogenase is that it is promiscuous in its redox cofactor utilization, and it can use either NADH or NADPH as a electron donor acceptor. So it's got kind of a few unusual features going from it. Okay, and this enzyme glutamate dehydrogenase, this is important for the generation of glutamates and also in the catabolism of glutamates. And this generation of free nitrogen in the catabolic reaction, this is very important for a urea cycle and kind of impinges on the catabolism of all other amino acids, kind of a lot of other, like amino acid breakdown in your skeletal muscle. The carbon part of it goes to carbon dioxide within your skeletal muscle for energy. The nitrogen then can be converted into glutamate or glutamine or alanine and be transported through the liver. Then reactions like this can make free ammonia, which can be converted into urea and gotten rid of. Okay, so that's what I'm trying to say is we're going to see this again on Wednesday as an important reaction in the urea cycle. Okay. Okay, so once we have glutamate generated, that can be a substrate for one of the other rare enzymes that can use inorganic nitrogen as a substrate, glutamine synthetase. So the glutamine synthetase, this is using ATP hydrolysis to get energy and using the energy of that high energy phosphate bond to make a new carbon-nitrogen bond to make the amino acid that has the two amine groups glutamine. The corresponding enzyme in glutamine catabolism doesn't make a new carbon-nitrogen bond, it makes a free ammonium as the glutaminase is cleaving off that ammonium to make glutamates. Okay, glutamate can also be a substrate for synthesis of the amino acid proline, and to do this, it's making the glutamate semialdehyde, which can also be a substrate for, not shown here, but the amino acid ornithine. Ornithine is an amino acid that is not present in protein, of course, but ornithine is an amino acid that is an intermediate in the urea cycle. Okay, so in order to get the proline, we first have to make this carboxylic acid into an aldehyde. This requires both ATP energy and reducing energy from NADPH to make the glutamate semialdehyde. This then can non-enzymatically cyclize into a structure that's essentially got the proline largely there. The bond between this carbon and nitrogen then is saturated using NADPH to make the ultimate proline structure. Okay, so then in the catabolic sense, we have to reverse the process, and it uses different enzymes. So in order to oxidize that carbon-nitrogen bond, that's using FAD as a redox cofactor. Then this is the same step, but to go from here to here, we're using NAD plus as a redox acceptor rather than NADPH. Okay, that glutamate semialdehyde, this then can be a substrate for a transamination reaction to generate ornithine, and that ornithine is an important intermediate in the urea cycle, so we'll just leave that there for the moment. Okay, what questions do you have? This is a lot, I know, but hopefully it's kind of calling back to things that we've seen before. Is that true a little bit? A little bit, okay. And yeah, and as we finish out the urea cycle and then the tetrahydrofolate stuff, we're going to get all the pieces coming together. Okay, so I think that we have talked about the synthesis of all of the amino acids that we are able to synthesize. Okay, and along with the synthesis pathway, we've described the degradation pathways kind of along with it. So what we'll talk about next we'll introduce some of the degradation pathways for amino acids that we can't synthesize ourselves, that we need to eat in our diet. And we may touch on some of the degradation of some of the non-essential amino acids as well. Okay, so all the amino acids, whether they're essential or non-essential, they can either be broken down into intermediates of the TCA cycle, into glycolytic intermediates such as pyruvate ultimately, or into acetyl-CoA or acetoacetate. Now, we make a distinction between gluconeogenic amino acids and ketogenic amino acids to distinguish between amino acids that can be broken down into either acetyl-CoA or the ketone body acetoacetate. So acetoacetate, this can, essentially not exactly the reverse here, but acetoacetate can make two acetyl-CoAs, so it's kind of the same thing. So an amino acid that's making acetyl-CoA, this acetyl-CoA can never contribute carbon to gluconeogenesis. And it's kind of you have to think about like where acetyl-CoA fits into metabolism and where our gluconeogenesis substrates are. So malate, oxaloacetate, that's like the important pool in the mitochondria of our gluconeogenic precursors. That oxaloacetate, because of the energetics of the TCA cycle reactions, that flows towards malate principally, and then malate leaves the mitochondria, can reform oxaloacetate and make glucose, right? We can also go through aspartate as well. But let's just consider, as shown on the cartoon from Marx, malate being the key gluconeogenic substrate. Okay, if we have the amino acid glutamate, for example, glutamate can be transaminated into alpha-ketoglutarate. So that's 5 carbons total in alpha-ketoglutarate. We're going to lose 1 carbon as carbon dioxide through alpha-ketoglutarate dehydrogenase. And that leaves us with 4 carbons that can then become malates and become glucose. Okay, so if we start with glutamate, we lose one carbon as carbon dioxide, and we can contribute the remaining 4 carbons to gluconeogenesis. With acetyl-CoA, the story is different. So acetyl-CoA enters into the TCA cycle up here, and the 2 carbons that enter into the TCA cycle, those are going to join with a molecule of oxaloacetate that's already there to make citrate. Then there's 2 decarboxylation reactions to make carbon dioxide, and we're back at the 4 carbon step, right? So if we put 2 carbons into the TCA cycle here, we lose those as carbon dioxide. We don't have any net increase in carbon into the TCA cycle that can go out as malate for gluconeogenesis. Is that conceptually clear? Right, because we can go, if we have pyruvate, we can go to oxaloacetate through the pyruvate carboxylase reaction. Acetyl-CoA, that can't go back. There's no like acetyl-CoA carboxylation to make pyruvate, or there's no reverse of the pyruvate dehydrogenase reaction. Okay, I think this slide I don't think is in your download, but I'll add it in with the quiz slides after class. Okay, this might be a good break time, but there's a minute left on the clock. Do you have questions? Okay, well, why don't we break a minute early and come back at 11 o'clock. Or 11 o'clock.
How are you? Didn't need to skip me. I'm just really focused. Super zoned in. Yeah.
You're so good at that.
It was good. How was it? Fun. Good. Your friend's birthday was fun. Did you go downtown? Fun. I love it. Yeah, so it was fun.
Roll off the top. Not that one, it's not so nice.
You've got more to do.
I know. What you do, Connor McDavid?
I got 130 cards.
What do you do? I was wishing people who might think. Just
I think so.
And then that's what the Kyoto says. I was like, but after they were taking people like yeah. I heard last year.
From college or high school?
High school. Yeah. But he's like